So let's go over choroideremia. It's some of the unique aspects of choroideremia which make it a good target for gene therapy. And the first is the genetics. And I'm sure most of you understand the genetics of choroideremia. So I'm just including this slide in case there's anybody who is new to this area. But this is an X-linked recessive disease, which means that the females are carriers. They're not necessarily affected, although they can have disease symptoms depending upon the amount of what's called X chromosome inactivation. Females have 2 X chromosomes, and normally one of the X chromosomes is inactivated randomly throughout the body. If it happens to be the normal X chromosome that's inactivated, the females can actually have symptoms that are very similar to what a full-blown choroideremia would be. But what happens is that the female transmits an X chromosome to each of the children, and the male children are the only ones that receive an X chromosome. So if they are unlucky enough to receive the X chromosome which has the choroideremia mutation, they will be affected by the disease, whereas a boy who inherits the normal X chromosome does not. And female children are typically carriers. So it's a recessive condition, X-linked recessive, meaning there is a lack of function of the choroideremia gene. It does not produce the normal Rep1 protein that the choroideremia gene does produce. And so this makes it a great target for gene augmentation therapy, meaning one can deliver a normal, theoretically, one can deliver a normal copy of the gene to the defective cells and deliver the missing function. And we know that the choroideremia gene is expressed in at least 2 different cell types in the retina: the retinal pigment epithelium cells, which are the nerve cells of the retina, and photoreceptors. And thanks to the long career and productive career that Miguel Seabra and others have had in this field, we know what the protein does. It prenylates Rab proteins, which helps get them to membranous compartments. So we know a lot about it. We know that we need to have this protein in those specific retinal cells for them to be able to work efficiently. The next step is, can we deliver it? And one thing which makes it difficult to deliver many genes is that they're too large. Well, fortunately for all of us, the choroideremia gene is a nice size to be able to fit within the reagents that we use to deliver it.